In this video, we'll learn more about lipids and how they come together to form cell membranes. The plasma membrane surrounds the cell and is responsible for separating the cell's interior from the external environment. Some important functions of membranes are to keep damaging materials out of the cell, allow entry of materials that are needed by the cell, and to facilitate the chemical reactions necessary for life. Since phospholipids are insoluble in water, they cannot interact with water to form hydrogen bonds with their hydrocarbon tails. The parts of phospholipids that do interact with water are the hydrophilic heads that can form hydrogen bonds with water molecules. Therefore, upon contact with water, phospholipids do one of two things. They either form structures called micelles, where the hydrophilic heads face the water and the tails face the interior of the micelle, or they form phospholipid bilayers, also known as lipid bilayers. In this image, you can see lipid micelles, which are spherical structures consisting of a single layer of phospholipids with their hydrophilic heads facing the aqueous solution and their hydrophobic tails facing the inside away from the water. On the other hand, phospholipid bilayers form when two layers of phospholipids align with each other with their hydrophilic heads facing the water or aqueous solution and their hydrophilic tails, whether saturated or unsaturated, face the interior of the bilayer, again avoiding water. Scientists often use these bilayers as experimental systems to study the movement of molecules across cell membranes. Artificial membrane-bound vesicles that are formed from lipid bilayers are called liposomes. Scientists also use structures called planar bilayers, which are lipid bilayers constructed across a hole in a glass or plastic wall that separates two aqueous solutions. Most of what we know about the movement of molecules across cellular membranes has been discovered by studying these experimental membrane systems. The left panel of this image shows an electron micrograph of how liposomes look in solution and a cartoon representation of the lipid bilayer formed within liposomes. Panel A in this image is a graphical representation of how a planar bilayer is used to study movement of molecules by adding solutes or ions or molecules to one side of the membrane. You can ask questions like, how rapidly can different solutes cross the membrane when different types of phospholipids are used to make the membrane? So the composition of these phospholipids? Or, for example, how rapidly would the solutes cross if proteins or other molecules were added to the membrane? Lipid bilayers are very selective of the substances that they allow to pass through. Small or nonpolar molecules move across these phospholipid bilayers relatively quickly. On the other hand, charged or large polar substances cross slowly, if at all. This image shows the selective permeability of lipid bilayers to certain molecules. As you can see, small nonpolar molecules can pass through pretty quickly via hydrophobic interactions with the interior of the lipid bilayers, whereas polar molecules and large molecules have a tough time crossing the lipid bilayers due to their chemical nature. Same is true for electrically charged ions, to which the membrane is impermeable. There are many factors influencing the permeability of membranes. These include the number of double bonds between the carbons in the phospholipids' hydrophobic tail, the length of the tails, the number of cholesterol molecules in the membrane, and the temperature or energy provided to the system. Since double bonds cause kinks in the hydrocarbon chain of phospholipids, These kinks prevent the close packing of hydrocarbon tails and thus reduce hydrophobic interactions. Phospholipids that have more unsaturated hydrocarbon chains have at least one double bond, and as a result, membranes that are formed from these phospholipids are much more permeable to solutes. Saturated hydrocarbon chains that are part of phospholipids in a lipid bilayer lack double bonds and thus kinks in their hydrocarbon chains, and as a result pack more tightly, leading to a decrease in permeability and fluidity of the membrane. The presence of long saturated hydrocarbon tails leads to stronger hydrophobic interactions between the tails of phospholipids within a membrane, reducing the permeability of the membrane. The presence of cholesterol within membranes also increases the density of the hydrophobic section, thus reducing permeability. In addition to this, membrane fluidity also decreases with decrease in temperature because the molecules in the bilayer start moving slowly, thus reducing the permeability of the membrane to solutes. To determine the impact of adding cholesterol to membranes, the Geer and colleagues performed an experiment in 1968 using liposomes. Their hypothesis was that cholesterol reduces permeability because it fills spaces in phospholipid bilayers. If their hypothesis was inaccurate, that would mean cholesterol has no effect on permeability. Their experimental setup involved constructing liposomes with or without cholesterol and then measuring the movement of a polar molecule, glycerol, across the lipid bilayers of these liposomes. Their prediction was that liposomes with higher cholesterol levels will have reduced permeability. When they collected their data, they found that the membranes that contained no cholesterol were more permeable to glycerol than were membranes with either 20% of cholesterol in them or 50% of cholesterol in them. This permeability increased with an increase in temperature, which is another factor that influences the permeability of membranes. Therefore, their hypothesis was supported, and they concluded that adding cholesterol to membranes decreases their permeability to glycerol. In addition to that, they also noted that the permeability of all membranes that were analyzed in this experiment increased with increasing temperature. In addition to being highly selective of what substances they allow to pass through them, membranes are also fluid structures. The individual phospholipids within membranes can move laterally throughout the lipid bilayer. These phospholipids can also flip between the layers, but they do that very rarely. The speed at which these molecules move within and across membranes also depends upon the temperature, the structure of the hydrocarbon tails, and the number of cholesterol molecules in the bilayer. This concludes our section on phospholipids as components of lipid bilayers. At the end of this section, I would like you to be able to answer the questions listed on this slide. Thank you.